Hi everybody, Professor Hobbs here. I am the CEO and owner of Serious About Science, and I have started these short series of videos to be able to help walk you through the basic science, to be able to do all of these really amazing, interesting science experiments at home without the need for expensive reagents or any type of fancy curriculum. So today's lesson is going to be focused mainly on how are we going to work with bacteria, what are some of the main portions of sterile technique, and the ways that you can start working with bacteria to be able to set up your own science fair experiments or set up your own bacteria unit in your home. There are a lot of ways that you can incorporate this either into your science fair curriculum, your homeschool curriculum, or even just supplementing your science curriculum that your students may have in the public school system. So last week I went over most of the basic items that are going to be needed, most of those being a nutrient agar that you can purchase online through Amazon, or a Luria broth, LB broth media. So normally when I do my media, you can utilize other glassware. I tend to focus on using these very small reagent bottles. Now mine are 250 milliliters. The main reason is that my pressure cooker is relatively small, so I can't do anything like a liter. So I tend to do more 250 ml and 500 ml of these glass reagent jars. So normally what I do is I will measure out, so for example, 25 grams of either my LB or doing my recipe that I have with my nutrient agar, and then I end up adding this to my bottle, and then I use a graduated cylinder to add distilled water ideally, but if you need to, you can use the tap to my bottles. And remember, you don't want to fill, I tend to err on the side of caution, and I only fill these up to about halfway high because they will boil over, and you 100% do not want that to happen when they're hanging out in your pressure cooker. So you want to autoclave these at about 121 degrees Celsius for about 15 minutes, or in your pressure cooker. Usually once that gets up to the boiling point and the proper PSI, let that sit for 10 minutes, come back and turn it off. And now before you end up putting these in, because we can't tell if things are sterile or not, we will need to purchase some autoclave tape. This is a special tape that turns colors when it's exposed to heat and moisture, and so we normally put that on top of our bottles before we autoclave them, and that tells us that we have autoclaved them. That's really the way that we make sure that all of our stuff is properly autoclaved. And so I normally will make up a larger batch of this, say 100 mils or 150 mils, and I'll put it in here, and I will put this in my pressure cooker. And then I will actually just pour this out into my plates. And because the inside of this is sterile, as long as I don't stick my fingers in it and I'm just pouring out of it, then I generally am not going to have issues with the sterility of my media. So one of the things that I do when I am working with media is I normally will have a dental lamp. Once again, you can find these on Amazon. Normally you can use any form of ethanol. I usually find some at Lowe's to be able to pour into this, and this is what I normally use for my sterile technique. So what I'm going to do with you all today is I'm going to show you how to pour a plate. I'm going to show you how to spread a plate, and I'm going to talk to you about kind of the basic things that as scientists we do in a lab when we are working with bacteria. So I autoclaved this a while back, but I warmed this up in the microwave. Big thing to make sure that when you put it in the microwave that the lid is kind of off, it's not fully sealed, so that you don't come back to exploded glass. The heat does need to be able to escape, but you don't want it to boil over. So normally we open it enough to where things can get out, but things aren't necessarily getting in. So I'm not actually going to light my dental lamp, but I am going to show you kind of how I handle it. So normally when you can touch the glass with a gloved hand comfortably, that's when you know it's reached the proper temperature to be able to pour. So that's why you notice throughout this I tended to come back and kind of feel it. Really, that was me trying to make sure for the proper temperature. We try to get this down to about 60 or 70 degrees Celsius. We obviously don't want to be sticking a temperature probe into our sterile agar. So because of that, we tend to use touch and feel. If it is too hot to touch comfortably with a gloved hand, it is not ready. Give it a few more minutes, or depending on how long you put it in the microwave, it might take a little bit longer. So normally, we are going to either pour or we use a pipette to be able to remove from here, but generally I do pour. So if I'm going to pour a plate, and when you ordered, you did not order the plates that are already done. You ordered either agarose, nutrient agar already made for you in probably a plastic bottle, or you made your own. Usually we try to do this with 2 hands, but I try not to let my lid touch everything because I'm trying to maintain the sterile aspects of it. So normally I loosen it enough to where I can grab it with 1 hand like this. Mainly I'm just trying not to touch anything. Then I will come over my dental lamp and I will move back and forth. I'm trying to sterilize anything that's coming into contact with my solution. If this is a plastic bottle, obviously keep in mind you don't want to melt the plastic. I have my Petri dish here. It is stored upside down, and I do that for a reason, mainly because it allows me to grab it like this. Then I can pour my solution in, and it really doesn't take very much. You're going to kind of want to move it back and forth, make sure there's no bubbles, making sure it properly covers the bottom. You are going to want to flame again. Quickly replace the cap. Now, for those of you that aren't as good with your hands, you have smaller hands, you're not as knowledgeable, or you haven't had as much experience with working with that, you can put the cap down. Special preference, you'll notice these lids are ridged. And the reason for that is so that we can actually store the lid on the side. So we don't necessarily want to put it this side down because we might pick up bacteria from our table. And then we don't want to place it on this side because we don't want any bacteria in the air to come and fall onto it. So the preferable placement is actually on its side, and it depends. You may not have one that very easily goes on its side and it might actually roll off. Happens all the time. So that's why normally in the lab we get really good about being able to use our hand to hold it and bring it back, minimizing the touching of things. So now this is going to end up firming up, and then next time I need to use it, I will stick it back in the microwave, warm it up so that I can pour another plate. If I need to add something to this like an antibiotic or any type of nutrient for my particular bacteria, I will go ahead and do that. So right now, this is going to sit here and I'm going to do my absolute very best not to touch it. It's really not going to take that long for it to solidify, and we want this to be as flat as possible. And anytime we mess with the solution while it's drying, it may get lumpy or get a ridge in it. And so we really try to keep it there at room temperature until that firms up. And we are going to actually move its position once it actually does get firmed up. So biggest things coming from that are making sure you are maintaining your sterile technique. I tend to utilize a dental lamp to flame the mouth of any bottle that I'm using. My bottles are actually glass. If you are plastic, you can still flame. I still flame. I just flame it a little bit less so that I don't actually melt the plastic. Causes kind of a problem when you try to put the plastic back on it. So just make sure you're very quickly flaming it. You are not holding it there and letting it hang out there to get super, super hot. It's just one way that we try to minimize the contamination between things. So those are how you are going to actually pour your plates. If you are either making your own nutrient agar or if you have purchased that through Amazon already made for you. Those are the 2 ways that you can do it. Once again, the other really easy way is to go ahead and order that already done for you. Obviously that makes it more expensive, but that is all of the different ways that you can do it. I do not order pre-poured plates. I make my own solutions, and then I just use a pressure cooker to autoclave my items, getting them to a high enough temperature and a high enough pressure to be able to kill anything that might be in there. Now, I have done the same thing to this. This is my LB broth. This I actually autoclaved at the other lab. But this one, because it's in a larger bottle, this does not have the agar in it. So it is just LB media. And this contains everything that any type of bacteria may need to survive. But it's in liquid form. So we will not always use bacteria hanging out on an LB plate. For us, actually, what we try to do is we try to use special tubes, culturing tubes, and I actually store mine. I pour my LB media in what we call 15 mil conical tubes. I put the cap on them. These are sterile tubes, and then I actually store these in the refrigerator and pull them out when I need them. These are 15 mil conicals, also available on Amazon. More expensive areas where you can buy them obviously exist, but Amazon turns out to be the easiest for those for me. So what you all are going to do, normally you are going to order a source of bacteria. Now you can either culture your own bacteria, say if you're going to go find a doorknob or something that you touch a lot to be able to collect bacteria. But if you are going to do an experiment that's utilizing E. coli, chances are, or another type of bacteria, you are going to order that. So I actually order my bacteria from homesciencetools.com. I do not get paid by them in any capacity. I just happen to use them and I find them to be affordable for most of my experiments. You will end up receiving your bacteria and it kind of looks like this. This is a special kind of tube. We call this a slant, mainly just because the agar was poured in such a way that it forms a slant when it's upright. And you will see that it's got stuff on top of it, and those little areas that you see that are on top of the agar are going to be our bacteria. This is a version of E. coli that is generally safe for us to play around with. We have a variety of strains. If you are familiar with E. coli, you will know that some strains are safe. We have E. coli hanging out in our digestive tract. Some strains can actually kill you and are sources of food contamination. Most of the time when you do receive a strain of E. coli, it's going to be a safer strain that's been propagated so that it minimizes the risk to the user. You'll notice as I'm touching all of these, I have gloves on, and I'm doing that to help minimize the impact on me. Once again, gloves really readily available online or on Amazon in a variety of sizes. So when you order your bacteria, it will come like this. Now the bacteria are hanging out and they are living here, but we want to be able to use them. And so to do that, we are going to want to move the bacteria from here over to our plate. And so to do that, you are going to want to purchase inoculation loops. These are available online. These are sterile inoculation loops. I prefer to buy the kind that you can throw away. It's just so much easier to deal with, and I'm less likely to contaminate other samples, especially not picking colonies. You will notice volumes ascribed to your inoculation loops. This is one of the really, really tiny ones. I do not have any preference for sizes. Honestly, order whichever is cheaper. But when they talk about those volumes, they're talking about the size of the circle at the end of the loop. It does not matter. I promise you will end up getting some amount of bacteria when you end up using these. So when you are going to end up doing your plate and you want to be able to move your bacteria from here to a plate, this one, because you are using sterile inoculation loops, you will not be flaming these because they are plastic, and when they come out, they are sterile. Now, I buy these super cheap online, and oftentimes my loop is on the wrong end. So it will say open here, but you do not want to open there because that is where my loop is, my circle. So in cases like that where you're ordering cheap reagents online, you are going to want to open on the end that does not have the loop. So I push that open. Now what I try to do is I try not to breathe on it. I try not to move around it. My gloves are not sterile. They're really just protecting me and minimizing my contamination process. So I still don't want to touch here. So normally, after many, many years of doing this, usually I fold it down like this. And then you are going to be very, very carefully removing it from its sterile container. Now, this is sterile. I'm going to do my best not to breathe on it. Keep it away from me because I do not want to introduce any bacteria here. So then with one hand, you are going to come in, you are going to put it in, and then you are going to just scrape, trying to get as much bacteria as you can on there. And you're just going to see some stuff. I do not recommend getting it up close and, don't shove it close to your face to try to see it. Just know it takes very little bacteria for us to be able to move our bacteria around. So just know that if you've put it on that area of bacteria, you 100% have pulled some up, even though you may not see it. So bacteria is very, very small. You have gotten some, believe me. So my plates, my plate has hardened. Now normally I want you to give this half an hour, but I have this one up here and ready. And like I said, I normally will try to use my one hand and pull this up, and then I'm going to start to spread my bacteria. So I'm moving my bacteria from that small tube onto here, and I'm going to show you with a picture how I'm spreading this. But I'm very gently, without digging into the agar, going to place that on top. Going to rotate this around 45 degrees. I'll give you a better picture. Then I'm going to put the top on, and what I'm going to do is I'm going to flip it upside down. I'll talk to you about that in a minute. Now, I keep my wrappers so that I can then put my gross, disgusting inoculation loop back in here and then put it over in my waste container because I don't want it anymore and I don't want it to move anything around. And that is how you are going to move your E. coli that you get into your tube onto your plate. Now you'll notice that I store these upside down. Really what happens is because there's a temperature differential here in our storage methods, we'll be going from say 37 degrees Celsius over to 4. And so because of that, we can develop condensation. If we stored this like this, condensation would occur up here and fall onto our plate, otherwise contaminating our plate. Not ideal. So generally we are going to store these here, upside down. If we look at them, I generally will look at them like this and then put them back down. So I keep mine stored like this just to minimize the amount of contamination that we can get from any condensation that we get here. You are going to want to label this. Every student I've ever met wants to write as big as they can all the way across here. I don't recommend that. I recommend getting a Sharpie and coming in and writing very nicely on the edge so that you don't cover this up and you make it a little bit easier to see. Now, I showed you how to spread, but it was a little bit harder, and so I'm going to demonstrate how I actually spread my bacteria and why we actually do it that way. So what I do when I spread my bacteria is I come over here and gently, I am only going to spread half the plate. Then I turn 45 degrees. I'm not going to do anything else with my loop. I'm not going to get a new sample. I'm going to keep it the same. Then I'm going to pull from here up and do half this plate. Then I'm going to do here. So in the end, I ended up spreading my entire plate. Now, why do we do that? We really don't know what the concentration of our bacteria is when we start to spread. So because of that, we have a lot of bacteria here. And sometimes when we get too much bacteria, it's hard to pick individual colonies. So because of that, we call it a lawn. We don't like that. We want individual colonies. And so what this process does is it takes this very heavily concentrated area of bacteria and then drags it out. So we're kind of diluting it and making less bacteria here And then when we do it again, we're getting even less bacteria. So what you'll find is that there's heavy amounts of bacteria here. And then as you go around the plate, you'll have less. And that is our method of minimizing and trying to dilute our concentration of bacteria across the plate. That is why we do that. Now, normally as scientists, we don't take our bacteria from an E. coli tube and then put it straight into onto our plate. Oftentimes what we'll do is we will take our bacteria from here. We will then put LB media in what we call these are my 5 ml culture tubes. And what's special about these culture tubes is that they are sterile, but they have a special lid that actually allows air to get to the inside, but minimizes the ability of anything to get inside of it. So when I culture bacteria or I'm trying to make more, I'm going to add 5 mils of LB to here. I'm going to take my E. coli and inoculate here. Usually I flame those, top on, top off. And then we actually have what's called an incubator shaker. So bacteria actually, when they're growing in this liquid media that we end up putting in it, they actually are a little bit too heavy for the solution and they'll end up falling out of solution, which isn't ideal. So what we do is we put this in a shaker. And so what that does is it literally shakes it while also incubating it. And that's really important because that allows us to actually get more bacterial growth in here, because when they fall to the bottom, they will end up actually getting too close to each other, and that will actually tell the other cells to stop growing, stop dividing. We are crowded, there's not enough food. And so that's one of the reasons why when we culture bacteria and trying to get a lot, we will add our liquid media to these culture tubes. We will put this in here, add it overnight in a shaker incubator, and it gets lots and lots of media in it, or lots and lots of bacteria. So this is what our media looks like when we don't have any bacteria in it. In here, it'll get really nice and cloudy, and it grows a lot. And what we will then do is I will then use the inoculation loop to move this sample over into my Petri dish. So that is not something, most people do not have a shaker incubator at home. We actually ended up building ours because it is actually a really expensive piece of equipment. You can do this at home if you want to culture more bacteria. You can put it in a windowsill to give you the proper temperature, a little bit warm, but you'll need to come and shake it around every once in a while to kind of get some movement in there. You're just not going to get as much growth as you would in a lab where we've maximized the growth. This shaking allows the bacteria not to hit each other and tell each other to stop growing, while also increasing the amount of oxygen that gets dissolved into our solution and really helps them grow nice, happy bacterial cells. So those are our 5 ml culture tubes. Once again, still order those on Amazon. Our lab would not be stocked without them. And so that is how we are basically going to handle the bacteria that we use for your experiment. So if you have either cultured your bacteria from around you or ordered from E. coli or ordered a form of bacteria online, you can buy these on Amazon. Like I said, I order mine from homesciencetools.com. There are other places online. Please compare prices. Some places really jack up the price, and I found that Home Science Tools is a little more affordable relative to other science supply companies. So what can you do with this plate of bacteria? Well, first of all, if you are doing this experiment, please pour more than one plate, and honestly, do 5 or 6 different samples. That's what's really great about bacteria in terms of experiments, is that you can do a lot of these experiments all at once in a short period of time. So what you can do with these bacteria is you can buy sterile paper discs. Once again, purchased on Amazon and a variety of internet sources. You can soak those in cleansers and put them on this plate. Now, if you're going to be doing that, you want to put them on this plate after you have put the bacteria down. Okay, so don't grow the plate, then put it on. Okay, big thing right there. After you have streaked your plate, put those discs on. I've had students run experiments where they've put, 409 or bleach, and they've put the discs on, and then they have incubated these. Your bacteria will grow at room temperature, albeit not the ideal temperature for E. coli. Put these in a sunny spot. If you want to get a little bit more warmth, yours is going to take about twice as long because you're using a little bit of a lower temperature. For me, I have an incubator that gets it to grow most of the time overnight within 24 hours. So science fair experiments with bacteria, you can end up doing those discs, sterile discs that are soaked in a cleaning solution. Then you're going to want to measure the distance of the zone of inhibition. So that's an experiment to do. You can also purchase antibiotic discs. Antibiotic discs are different concentrations of antibiotics that you can place on here, also measuring the zone of inhibition. Really great because it gets you numbers. Scientists, we love our numbers. And that's something that you can very easily measure. And then, like I said, when you do multiple plates, you can get 3 and 4 sets of data. Then you can get averages, you can get statistical analysis of those, and those are going to look really great on science fair projects. We normally try to do things in triplicate. More is always better. But bacteria is a really great tool to be able to run very simple experiments with minimal risk. I've done this with minimal cost in terms of reagents, all easily accessible. Some of the other things that you'll need, small little scale, purchased online. Make sure you purchase some autoclave tape. You may not need the autoclave tape if you end up purchasing your reagents already pre-mixed. But if you are pre-mixing, you are going to need those. The other item that you're going to need are things like weigh boats to be able to measure out your powdered solutions. And so things that are going to help with your sterile technique are going to be things like this dental lamp, available online. When you're done with the dental lamp, make sure you put that on nice and tight. Get rid of any flame. You can fill this with any alcohol source to be able to light that for later. So in this particular series, you were able to see all of the basics of our culturing of bacteria, how we are going to move the bacteria around, and how you can use it to make a really awesome science fair experiment, or just stop the boring science. Join us again for more information and more tips.